Start the recording. Share the screen. Boy, that's slow. There, do you guys see the screen now? Hello?
Yes,
You guys are not.
yes.
Yes.
Maybe it's a good thing that this came up. I wanted to remind you that there is an extra credit assignment for most weeks, meaning I think it's week 2 through 10. You can do the extra credit, what do I call it? Extra credit study? Yeah, extra credit study for the week number. So this would be extra credit study 3, just to get together with another student and study some material in the class. And then one person needs to submit what you studied, how long you studied, when you studied, and the names of everyone in the group. You do need full names, meaning I can't do it from just the first name. I suppose I probably could do it from the last name, but put the full name in. And then I'll get you an extra credit point. So you can do that every week. Just click on the EC Study 3 link for doing it this week. I think that's this week. Yeah, this week. You only get 1 point for each week, so you can't wait until the end and then do them all. And it has to be submitted and the study has to be done here. But over Zoom, it's pretty easy to do. If you have a phone, you could do it over the phone. It'd be best if you could do it with a visual phone, or you could do it just with sound over the phone. And it is a good way to study. And usually your study buddy will notice you're weak on something and then ask you further questions, which will strengthen your expertise in that area. All right. Another thing I need to mention is that you do need to send me your infectious disease topic so that I can approve the topic. You can simply not do the project without getting it approved. There was one person this term where I pointed out, that's not an infectious disease. So like I said, please clear it with me. Anything else I need to discuss? Oh yes, there is another thing I need to discuss, and that is I have been grading some of the assignments I got. Let me stop sharing the screen and I'll make certain I get it right. You should not be seeing the screen now. I'll double check that. And I'm going to come and show you the grades, and that's just an easy way for me to look and see which lab I did. I think it was lab 2, but let me look at the grades and I'll be certain of that, where I graded it. And that one's completely done, and I'm beginning the next one. So worksheet, yeah, lab 00 was completely graded. Lab 1, I think I graded that earlier, and lab 2 I just recently graded, and I'm working on lab 3. My goal is to have lab 3 maybe done tonight, or at least by tomorrow, and then lab 2 and lab 3, if you want to make corrections, they have to be done by this Saturday. For lab 1, you see when the corrections had to be done. Sorry, I just don't remember. The corrections had to be turned in September 30th. So I must have graded that last week. All right, any questions about any of that? And remember in the lab, if I say something's wrong, you can make corrections for full credit by the new due date. In the lecture worksheets, If you don't know the answer, you should look it up because you will not get a second chance in the lecture worksheet. And I decided to go that way because the lecture generally is only asking for 1 or 2 questions. Any questions or comments about that? All right, if not, let's begin. Am I showing you? I probably am not because I'm showing grades here. Hopefully I'm not showing you the screen, showing what's on my screen.
We do see you.
Great. All right, so let's begin sharing the screen again. And I shut down the grade book. So, all right, let's take a look at what we're doing. We're doing lab 4 stains today. Now remember, in the lab, when I'm done talking, you guys can work on the lab and ask me questions. That's why I'm sticking around. When the last student logs off, I will log off, but at least initially, I will be there to answer questions. I don't have this one marked for teacher's edition, so let me see if that's the right one. Yep, that's the right one. Lab 4, staining techniques. We'll be discussing simple, differential, and special stains. You should read some material from the textbook, and it's a lot of material, but they're each one is only a couple of pages. The size and shape and the arrangement of bacterial cells. You should read about that. Bacterial cell wall, simple stains, differential stain. Of the differential stain, there's 2 of them we're going to discuss, the gram stain, the acid-fast stain. You should read about those. And then lastly, you should read about special stains. And there are 3 main types. There's actually lots of special stains, but 3 we're going to talk about. A negative stain for a capsule, an endospore stain, and then flagella staining. And you can read about those. There are some supplemental materials, a video clip, and a virtual lab. I will check the virtual lab when we get to it to make sure that there's a virtual lab. I think there is a virtual lab on the staining. Upon successful completion of this laboratory, you should be able to prepare bacterial smears for staining, understand the principles and the procedures used when performing simple, differential, and special staining techniques, be able to interpret the results for a simple stain, a differential stain, and a special stain. And then lastly, define the following terms. What's a simple stain, a differential stain, a special stain? What is a coccus, bacillus, a spirillum? What's peptidoglycan? What's a gram-positive cell? What's a gram-negative cell? What's an acid-fast stain? What is a primary stain? What is a mordant? What is a decolorizer? What is a counter stain? What is a capsule? What is an endospore? And what is a flagella? Because of their extremely small size, bacteria are nearly transparent. Therefore, it is necessary to stain them before they can be observed under the microscope. Preparing a bacterial smear is just by dropping sterile water, usually 1 drop, on a clean slide, and then adding a sample of bacteria. And then you smear that bacteria over the surface of the slide. And that is all a bacterial smear is. It contains, the bacterial smear contains no stains. And then you let this usually air dry and then heat fix. And the purpose for air drying and heat fixing is to attach the cells to the glass of the slide so they don't wash off. In the staining procedure, which most staining procedures have a water rinse, and that would just rinse off all the cells. if they were not air dried and heat fixed. A simple stain is where you simply use one dye to stain the cells. And it says one basic dye, but it could be a, what's the other one called? I forget now, negative dye, is that the term? No, acidic dye. It's actually, it doesn't matter. It's just one dye. It doesn't matter what it is. It's usually a basic dye, but it could be an acidic dye. A differential stain usually uses 2 or more dyes, and a differential stain provides additional information about the cell. And a differential stain we're going to talk about is the Gram stain. It's the most commonly used differential stain, and probably the most commonly used stain used by microbiologists. Lastly, there are special stains, and they're used to stain a particular structure of bacteria or of a cell. Any questions about any of that? All right, so you should know a cocci is a cell which looks like a sphere, and they can be in different cellular growth arrangements, also called different growth arrangements. And you can have singlets, and that would be a single cocci. And then there can be diplococci, 2 of them. There can be tetrads, 4 of them. There can be staphylococci, and that's a clump of cocci. And staphylococci actually means it looks like a clump of grapes. And if you look at that, that kind of does look like a clump of grapes. And then there's also streptococci, where they're in a chain. For bacilli, there are some of these arrangements, but there are a few of them. Once again, there are singlets or single bacilli. There are diplobacilli. And there is a streptobacilli. Any question about that? For the bacilli, there is a normal bacilli, and it can be square at the end or pointed at the end. Most of them are rounded like this. There can also be a shape in between coccus and bacilli, and they will have rounded ends. And simply if you shrink the bacilli shape, you'll get a coccobacilli. And it sort of looks in between a cocci and a bacilli. There can also be pleomorphic bacteria, and they'll take on different shapes. This is sort of pleomorphic. This one's pointed at one end and fat at the other. That one's kind of somewhat slight S shape. I don't know what you want to call that one. It just looks different than that one and that one. Pleomorphic, meaning the cells take on different shapes. Most bacteria are not pleomorphic. Most bacteria have one shape for their cell, and that would be mostly a cocci or a bacilli. And even the bacilli, all of them will either be rounded ended or pointed ended or square ended. Okay. Now it's true with some of the coccobacilli that they can be a little bit pleomorphic, but they'll always be a combination of looking in between a cocci and a bacilli. And then I don't want to call this a spirilla, a bacilli. I want to call that a spirilla. And that will be a cell which has a bend or 2 in it. And these look like S's. And those we call spirilla, or a singular spirillum. Okay, any question about that? So you should know those terms. All right. Of the differential stain, the Gram stain is the most important. Now remember, a differential stain allows you to see differences between different cells. So like if I had a slide that had 2 different species on it, I could stain it with a differential stain and then see differences between the cells. They may have a different color, for example. If I'm using the gram stain, the gram positive cells will stain different color than the gram negative cells. The gram stain was invented by Hans Christian Gram. And you don't need to know when he did it, in 1884. He developed this as a differential stain so that he could distinguish in a patient the bacteria cells that would stain different from the patient cells. So he intended it as a differential stain, but microbiologists started using his stain, and they found that it can be used as a differential stain to show differences between different bacteria, meaning the gram positive and the gram negative. Okay, you didn't develop it that way, but that's the way it became. And you should know that this stain is developed by a person. And that's why when we use the gram stain, you should always capitalize that G, because gram comes from Hans Christian Gram, a person. Any questions about that? All right, the gram stain uses crystal violet first as the primary dye. And if the bacteria retain that primary dye, meaning the crystal violet, we will classify them as gram positive, and they will look purple in appearance. If the bacteria do not retain the crystal violet, they will not look purple in appearance, and we will call them gram negative. They will appear pink from the gram stain, which will remove the crystal violet from these cells. And then we stain it with safranin, which is pink or pinkish in color. Okay. And here we have a typical gram stain. And it looks like we have many different cells in here. Like there's a gram positive coccus, a gram positive rod. Someplace there's a gram negative rod. There's a gram negative rod, or a gram negative bacillus, same thing. And then there's a gram negative spirilla. Okay. Any question about any of that? The gram reactions are based on the ability of the bacterial cell wall to retain the crystal violet in the procedure. The gram positive microorganisms contain a thick peptidoglycan layer in their cell wall, whereas the gram negative bacteria have a thinner peptidoglycan layer. Okay, so gram positive, thick peptidoglycan layer, gram negative, thin peptidoglycan layer. And the gram negatives also have a lipopolysaccharide portion to the cell wall. We'll talk about that later. Gram positive cell, a thick peptidoglycan layer, and you note there's many, many layers there. The gram negative cell shown here, let me get that down a little bit. This is the cell wall portion right here. That's the plasma membrane, the greenish portion, plasma membrane. The gram negative, this is the cell wall. And it does have the peptidoglycan. It's just thinner than the gram positive cells. And the gram negative cells also have a lipid layer, bilayer, actually, we'll talk more about that, shown here. And we'll talk more about that. That's called the outer membrane of the cell wall. Some gram-positive bacteria lose their ability to retain the crystal violet dye, especially when they're old, and they are easily decolorized. Sometimes they're described as gram variable, although they are gram positive, and they appear as though they are a mixture of gram-positive and gram-negative bacteria. These cells usually are mycobacteria that are, I guess, gram variable. And when you stain them, like I said, you can see gram-positive cells here and gram-negative cells there. Let me blow this up a little bit, get this over here, and ask you, what do you know about the difference between the, oh shoot, why is that not working? Looks like I'm, let's do it down here. What do you notice about this cell, for example, and maybe that cell, or that cell, especially that cell there? What do you notice about the difference between that cell and that cell, besides one's gram-positive, one's gram-negative? You know, if you're wrong, so what?
The shape?
Yeah, they do have a different shape, but that's not what I'm getting at. There's another difference in the appearance of the cells. Say that cell here and that cell there. What do you notice between the difference in the appearance of those cells?
Different color?
Yeah, one's gram-negative, one's gram-positive. What's another difference? Keep going.
The clarity of them?
Yeah, this one is sort of ghostly. It's not very clear. The boundary isn't very sharp. And this one is not ghostly. The boundaries are very sharp. What happened is this is probably a new cell. And so the cell wall and cell membrane is intact. And so the cell stained sharply. This cell is a ghost because it's an old cell and the cell is decaying. The cell wall is not fully intact anymore. And usually, whenever I've seen a student who stained the cells and they got a gram variable appearance, you know, this isn't gram variable if that's a rod and that's a spirillum, those are two different species. But in the case of this one and that one, and that one and that one, it's gram variable. But in the case of all the gram variables I've ever seen, I asked the student, how old are the cells when you did this gram stain? And every student who had the gram variable, even if it was mycobacterium, they always told me, oh, it's 2 weeks or older. Usually it was 3 weeks or older. And I said, you can't do that. If you're doing the gram stain, the cells have to be 2 days or less old. Otherwise, the gram stain may not work properly. Okay? And why is if the cell is deteriorating, the cell wall is deteriorating, the crystal violet may wash out of it. And it's simply because the cell wall is not intact. Okay? Any question about any of that? I'll talk a little bit more about the, you need to do the gram stain on young cells in a little bit, but let's go on and say that every case where I've ever looked at the gram variable results, it was only because the cells were too old. And if the student ever used young cells, they always got usually gram-positive cells because the mycobacterium are gram-positive. Okay, any question about that? All right, so the gram reactions are based on the ability of the bacterial cell wall to retain the crystal violet. The gram-positive cells have a thick peptidoglycan layer, and that thick peptidoglycan layer retains the crystal violet, while the gram-negative bacteria have a thinner peptidoglycan layer, and that thin layer of peptidoglycan does not retain the crystal violet in the gram stain. I think I've already talked about that, so let's move on. Yeah, The mycobacterium are the hardest ones to stain with a gram stain, and that's because mycobacterium have a lot of mycolic acid in their cell wall. That substance, mycolic acid, is a lipid, and it prevents the dye, which is water soluble, from getting into the cell. And so the mycobacterium do not stain well, but they generally do stain gram positive if you use young cells. Okay. Now I've only tested that on Josh. I guess I've only tested it on 2 species. But all of those cells that I've ever tested, that's the case. And I guess that's true. I haven't tested a large number of species of mycobacterium. Like, for example, Mycobacterium leprae, I've never grown. It's a human pathogen, and I've never grown it. Some tips to prevent gram variable staining. During the smear preparation, do not use old cells. The cells should only be 2 days old or younger. Prepare a thin smear. If you have a thick smear of cells, meaning cells on top of each other, it's hard for the dye to get into all of the layers of cells. And it's also hard for the crystal violet to wash out if it should wash out of all the different layers. Heat fix the bacteria lightly only after the smear has been air dried. If you don't air dry the slide and then you heat fix the cells, there will be too much moisture in the cell, and that'll put a hole in the cell. And so the crystal violet may come out of the gram positive cells. If you use too much heat to heat fix the cells, you'll actually start melting the cells, and that can disrupt the cell walls. Use obviously fresh staining reagents when you're doing the gram stain to help with the gram stain. Do not over decolorize, but not under decolorize. You rinse, use the decolorization step for as long as the dye is running. Once the dye stops running, stop decolorizing, and then immediately rinse the slide so that the decolorization agents are washed off the slide, because even a trace of it left on will work. And if you decolorize for too long, you will remove crystal violet from cells where the crystal violet should be retained by the cells, meaning you can remove the crystal violet from the gram positive cells if you decolorize too long. And then inversely, if you decolorize not long enough, you will not remove all of the crystal violet from all of the gram negative cells. All right, any question about the gram stain? Okay. An acid-fast stain is another differential stain, and that can pick up some bacteria that are in the families of Mycobacterium and Nocardia that have a waxy substance called mycolic acid in their cell walls. These bacteria are able to resist the acid alcohol decolorization. And so we call these cells acid-fast bacteria. They take up the acid-fast stain because the acid stain goes in the mycolic acid, and then it doesn't wash out. And so these are the acid-fast bacteria. Other cells of other families, not including the Mycobacterium and Nocardia, the acid-fast stain will wash out of them. Okay, any questions about that? All right, we're not going to go into great detail with the acid-fast stain, although you're going to read about it. Acid-fast bacteria stain pink from the dye. Carbol fusion. And then the acid-fast negative cells stain either greenish or bluish from the counter stain. So acid-fast negative cells, acid-fast positive cells. Any question about that? Let me warn you, if you're doing the worksheet for the lab, there is a question to ask you what cells are acid-fast positive and what cells are acid-fast negative. And if you can write it in with the arrow. You can also write it in as a description saying that, I don't know, the first arrow is acid-fast positive. The second arrow on the right is acid-fast negative. Any question about that? All right, that's enough for this differential stains, which, remember, they make the cells look different or stain differently. And this stain cell looks differently than that cell there. And this is a differential stain. There are also special stains, and they are stains that are developed to specifically stain a specific structure or part of the bacteria or of the cell. One in particular is the negative stain, which stains for a gelatinous substance called the glycocalyx. And if the cell has a glycocalyx, it will be attached to the cell wall. And if it's firmly attached and you can see it under the light microscope, we call it a capsule. There's also a slime layer, which you cannot see under the light microscope, and it's mostly water, but it is another glycocalyx, okay? And it's attached loosely to the cell wall, or at least looser than a capsule. So if you see a glycocalyx under the microscope, you're looking at a capsule. So here we have a bacteria. There's the cell membrane, there's the cell wall, the yellowish one. And this cell made a capsule, which is the green layer or area outside the cell wall. Production of a capsule is important because it can be associated with an organism's virulence or its pathogenicity. And so, like, for example, Streptococcus pneumoniae, or let me see if I can remember, Streptococcus pyogenes. Is that right? Yes. If it has a capsule, it will be more virulent and be more likely to cause disease in a patient. Okay. And as you can guess, Streptococcus pneumoniae causes pneumonia, and Streptococcus pyogenes causes strep throat or sore throat. The reason why the body has a hard time throwing off these infections if the cell has a capsule is the capsule is protecting the bacteria cell from the host immune reaction. I'm trying to see if I've got a picture of that. I don't think I do. And that is why Streptococcus pneumoniae, an infection in the lungs, is so dangerous, because obviously pneumonia is dangerous anyways, but the patient can't throw it off. And so their immune system can't throw it off. And so they would need antibiotics usually. The same thing with strep throat. The bacteria have a capsule around it. And so humans are not good at throwing off or killing off the bacteria causing the strep throat. And that's why strep throat oftentimes just gets worse with time. And then the patient has a sore throat for a month or more. And then they go in and see the doctor and they'll do a test and find out they have strep throat. All right, any question about any of that? Now, if the bacteria have a capsid or capsule, it tends to make the bacteria grow mucus. And so these colonies here, which look mucus, it's because they have a capsule and it makes them look mucus. When we do the capsule stain for Klebsiella pneumoniae, it can look like this. The capsule stain stains the background of the cells, meaning the background of the slide, and stains the cells also, much darker in this case, but it does not stain the capsule. But that allows us to see that these cells have a capsule around them. Any question about that? Oh, and why it's called a negative stain. If you know anything about old photographs, they used to have a negative, and digital cameras, of course, don't even have a photograph, but there is no negative anymore. The negative looked like this, where the person in the background would be dark, and then there would be other regions which would be light. I'm talking about a black and white photograph. And it would be the opposite of the way a person looks. Like my hair would be all white in the negative, and then my skin would be all dark. Okay, and that's why this is called a negative stain. Any questions about that? And is there a difference between the negative stain and the capsule stain? If it's used to find the capsule, those terms are used interchangeably. Any questions about that? A negative stain does not have to be used to look for a capsule. You could use a negative stain to stain the cells for other reasons, in which case the two would not be synonymous. But in this lab, the two terms are synonymous. Hopefully that wasn't too confusing. Another special stain is an endospore. You can have a special stain where the stain stains endospores. Endospores are metabolically inactive cells, and they're very hardy structures that are produced by some bacteria as a defensive strategy when the environment is becoming unfavorable for the cells to grow. Most of the bacteria that form endospores belong to two families, Clostridium family and the Bacillus family. When the environment becomes favorable for growing, the bacteria will grow and they will not form the endospores. But when the environment decays, the endospores will be formed by the cells. Endospores are not metabolically active, and it's true they have some metabolism, but mostly they don't have any metabolic activity, meaning it's very light, very little metabolic activity. And the purpose is just to keep those cells alive until the environment becomes favorable. And then the endospore will germinate and the bacteria will return to their normal metabolically active state. And we have a name for that. We call it the vegetative state. Any questions about that? You'll need this table to answer some of the questions. It's giving you some of the different species of bacteria that produce endospores, and then the diseases that they cause, like Bacillus anthracis causes the disease anthrax. Any question about that? All right, the Schaefer-Fulton endospore stain, often just called the endospore stain, it's named after the people who invented it, uses malachite green as the first stain. And malachite green will stain the endospores, and the endospores will retain that malachite green, whereas the other cells that are not endospores will not retain the malachite green, and they'll take on the color of the counter stain. The counter stain is usually safranin, so they will stain pinkish the non-endospore cells, and then the endospores will stain green or greenish. Here is a free endospore. It's outside of the cell. It's called an endospore because it grows within the cell. And you see this endospore there between a red portion here and a red portion here. And there's another one there, although the arrow is kind of hiding it. And here's one here, although it's very small on this end. It's called an endospore because it grows within the cell. This red area is the parent cell, and it's making the green endospore. Okay, any question about any of that? Let's shrink that down. We also have a special stain that we call the flagella stain, and it stains the flagella along thread-like or whip-like appendage that's attached to the cell surface. And the flagella's purpose is to provide the cell with motility. If the cell can move, locomotion, and it's a bacteria cell, then it has to have flagella, because that's the only way that bacteria can at least swim and move. Any questions about that? All right. There is a question in the worksheet that asks you to give the name of the bacteria flagella. And you can use this table or whatever this is called for getting the name. Let me warn you that if there's more than one flagella type there, it's probably because the cell is making the flagella. For example, if it is an amphitrochus bacteria, you will see some cells are amphitrochus, but there will be some cells which are lophotrochus because they haven't made the flagella yet on the other side, right over here. And then there will be some cells which are monotrichous because they are just beginning to make the flagella. And so far, they've only made one. And when you see a mixture and you need to say what type it is, always give the name for the highest number of flagella. Any question about that? All right. Flagella are too thin to be seen by most stains. And so we have a special stain. And what it does is it coats the flagella many times so that it's coated enough so then we can see the flagella. And here we have a picture of cells having flagella. All right, any questions? So you should know the terms. And go ahead and do the worksheet. Let me look on the worksheet. Laboratory 4 to make sure there's a virtual lab there. Remember, if you're doing the lab module, to only turn in the worksheet. Somebody in one of the earlier labs turned in the lab module, and it was so big, I don't think all of the file transferred. Another reason why I don't want it is the lab module is so big that it will fill up my Canvas website, and I'd rather not that happen. So go ahead and do procedure 1, preparing smears for staining. We've got a video on preparing the bacterial smear and then heat fixing the bacterial smear. And remember, when you're making a smear, you're not working with any dyes, not yet. Now, usually you stain the bacterial smear after that. So when you're doing a simple stain or a differential stain or any other stain, you usually make a bacterial smear before you apply the stains. A simple stain, remember, is just staining with one dye. And we have a little video for watching on simple stains. Read through the simple stain and watch the video. And there's a picture where the different cells take on the same color, but you can notice some differences between these cells, and that's because they have a different shape. They may have a different cell arrangement as well, a growth arrangement, cellular growth arrangement. And then answer the questions. And then do the procedure or read the procedure for doing the gram stain. Watch the video on the gram stain. And then we have a virtual lab on the gram stain. The virtual lab uses different decolorization step than we're talking about. So the bacteria will be a different color. Is that what we're going to say? I lost my place now. Oh, no, it's a slightly different procedure. Where did I have that? It's on the worksheet. I'm not looking at the worksheet. That's the problem. Let me close the syllabus down. Come back to the worksheet. So that's not in the lab module. It's on the worksheet. But the virtual lab uses a different decolorization solution than we discussed. So the decolorization step in the virtual lab is longer than we're discussing. The procedure here is the procedure that's normally used. You use and apply crystal violet for 1 minute and then wash it off. And then you flood and smear the smear with iodine for 1 minute, wash it off. And then you apply the alcohol acetone decolorizer. And there are different decolorizers. And this one, you don't let it soak for only 5 seconds. The best thing to do is to hold up the slide. Let's see if this will show. Hold up the slide, add your decolorizer, and when the running of the dye stops, stop adding the decolorizer, and then immediately rinse with distilled water to wash off the decolorizer. In the virtual lab, it's longer, the procedure they're using. They probably are only using alcohol, and so you need to decolorize longer to get off the crystal violet. Once again, if you simply stop when the decolorizer stops running the crystal violet, then that's a good way to do it. But it takes longer in the virtual lab. Then you flood the smear with safranin, and that will stain the gram-negative cells pinkish. And then rinse with distilled water, and then look at the slides. under the microscope. And here's the procedure here. Application of crystal violet, all the cells stain purple. We then apply iodine, all of the cells stain purple. The iodine does intensify the dye, so these will be a stronger purple as well as a darker purple. A little less blue, meaning more purple color from the iodine. And then we have the decolorization step. The gram-negative cells will stain clear after the decolorization step. The gram-positive cells retain the crystal violet, so they're still purple. We then add safranin, and the gram-negative cells will be pink in color. The gram-positive cells will be purple in color. And then answer the questions in this table here. We stain E. coli and Staphylococcus aureus. Tell me if it's gram-positive or gram-negative. And you should say gram-positive, gram-negative, not positive or negative. And then give me the cellular morphology. That's their shape of the cells. And then answer the question after you apply the primary stain, that's the crystal violet, to the Staphylococcus aureus, what's the color of the cells? And the color of the cells, at least of the gram-negatives, will change in this procedure. So answer those questions. Then do the acid-fast stain, procedure 4. We have a video clip to watch. The acid-fast stains positive will be pinkish. The acid-fast negative cells will stain either green or blue, depending on what the counter stain is. Any question about any of that? Read through the procedure. Here's the question, which the question is right up here. Seen below are 2 smears. Interpret whether the organism denoted by the arrows are AFB, meaning acid-fast positive, or acid-fast negative cells. And I see why some people don't see that this is a question. I should put a question mark there to make it clear that that's a question. And you can write it here if you want, or you can just describe it, saying that the top cell on the left is either acid-fast positive or acid-fast negative. Okay. And then do that 4 times. Okay. 1, 2, 3, 4. Did it right. Okay. All right. And then do the special stains. Once again, we have video clips on the endospore stain and the negative stain. We have a picture here of a stain, which is either an endospore stain, a negative stain, or a capsule, not a capsule, flagella stain, sorry. And so you need to figure out what they are and then answer the questions. So A is a Schaefer-Fulton stain, and it uses what as the primary stain? So what's the name of the primary stain? Answer that. And then ultimately dyes what structure, which is produced by some bacteria to aid in their survival. So answer the questions and then submit the worksheet. And then at the end of this worksheet, we have 2 case studies. Read through the case study. Here's the lungs of a patient, and they have something going on right here. And then somebody did a sputum smear and stained it with the Zeal-Nielsen stain. You can look up what that does. And we can see differences between the bacteria and other bacteria staining in the cell, like maybe right there and there. And then these are probably host patient cells of the host. So what you need to do on these case studies is figure out which bacteria species is causing the disease, which is described in case study 1. And then give me the name of the species. I need a species name. Okay. And then read case study 2. And it's got some information for you. Here, I think they mentioned this is the diabetic. Yep, a diabetic patient. Soft tissue necrosis, and we have a picture of it right here. And then they did a gram stain. There's the gram stain. And then they did the Schaefer-Fulton stain. And from this information, you should be able, and the information in the case study, you should be able to determine what species of bacteria is causing this infection and name the species. Okay? All right, that's it for the lab. Get started. I will be here until the last student logs off, or until 8:20. At 8:20, I'm logging off. All right, get started.